Greetings, students. All right, so let's talk about how the cell cycle is regulated. So just a reminder, here is the cell cycle. It's 4 phases. So the 3 phases of interphase are G1 phase, S phase, and G2 phase. And then after interphase, the cell will go ahead and divide, and that is M phase or mitosis. And then the 2 resulting daughter cells start the whole cycle all over again. So the cell cycle is really complex and it's really carefully regulated. Let's think about how that works. So the way that the cell cycle is regulated is with a special group of proteins called cyclins. So the phase that the cell cycle is in is determined by the amount of these special types of proteins. They're called cyclins because they're regulating the cell cycle. Cyclins, cell cycle, see it there? So what cyclins do is they bind to and activate specific kinases called cyclin-dependent kinases, or CDKs. And then these activated cyclin CDK complexes can go ahead and phosphorylate other proteins. So they can add phosphate groups onto other proteins, and that can change the activity of those proteins. And that is what literally makes the cell go from one cell cycle to another. It's just these cyclins that are going to bind to CDKs and activate them, and then this kinase, this CDK, can now phosphorylate whatever its target protein is, and now you can see this target protein, because it's been phosphorylated, its shape has changed, so now this protein is active, and it's going to do whatever that protein does. Okay, and then given enough time, what happens is these cyclins eventually, they have like a really fast expiration date, so they will break down. And so all the time your cell is cycling between the amount of cyclins they have. And that's how cells determine what phase of the cell cycle that they are in. So we can look even more specifically at a graph that shows us this process. So here we have on the x-axis, this would essentially be time. So we're progressing through the cell cycle from G1 to S to G2 to M. And then on the y-axis, we have concentration of different cyclins. So we can see in G1 phase, we have this steady increase in the G1 cyclin. Makes sense. In order to get to S phase, we have this big increase, and once it hits its peak of this G1S cyclin, that is what starts the cell to kick off S phase. Similarly, we have this other cyclin, S cyclin, that you can see ramps up throughout S phase, and then M cyclin, it's going to ramp up until it reaches a peak at the start of M phase. Don't worry about like, oh my gosh, I need to memorize this graph or anything like that. No, not at all. It's more this idea that you have these big increases and decreases in these different cyclins, and they correspond with the phase that the cell cycle is in. So the key idea here is that the concentration of specific cyclins is going to tell the cell what part of the cell cycle to be in, because those cyclins bind to and activate kinases, CDKs, which can then cause like a cascade, a domino effect that activates a bunch of other proteins within the cell. So in fact, you can actually do an experiment, and scientists have done this experiment, where they take a cell and they introduce a specific cyclin CDK combo, and they can see that just doing that on its own is enough to push the cell to the next phase of the cell cycle. So for this experiment, imagine you have an unfertilized egg and it is stuck in G2 phase. It is just staying there waiting. It's going to be in G2 phase forever. It's not ready to go to mitosis. It's stuck. But what scientists did is they took these stuck cells and they injected them with this special protein called MPF. So MPF is actually a combination of a cyclin and its CDK. MPF stands for mitosis promoting factor. So can you guess what happened when scientists did this? So all they had to do was inject this MPF, and all of a sudden this cell started to go through the cell cycle and it started to enter mitosis. And it happens like right away. All of a sudden the cell is getting the signal, mitosis now. It doesn't matter if the cell is not ready. The presence of that MPF is enough to push the cell into the next phase of the cell cycle. Okay, so back to a regular cell that's just normally progressing through the cell cycle. So, like I mentioned, you have these cyclins and CDKs that are really important for a cell going from one part of the cell cycle to another. One thing that these cyclins and CDKs do is they regulate checkpoints. So throughout the cell cycle, the cell actually will pause and be like, hey, am I ready to go to the next phase? So for example, before entering S phase, there's a checkpoint. So remember, S phase is when it stands for synthesis, because we're synthesizing DNA. The cell is going to copy its DNA and get ready to make two daughter cells. So before that happens, the cell actually checks to see if there's any damage to its DNA, maybe like UV radiation that has caused some DNA damage, like some breakages. And if there is damage, the cell will try to repair it. The reason for that is the cell doesn't want to start to copy its DNA if its DNA is messed up, right? Because that's how you can get mutations. So this is an example of a checkpoint. The cell's going to check itself before it moves on to the next cell cycle and wrecks itself, right? So you want to make sure that things are all good before you move to the next phase. So there's a lot of checkpoints, but really three big ones, and you can kind of logic your way through what they do based on where they are. So the G1 checkpoint, so called because it's in G1, and it's right before S phase when the DNA is replicated, so it's important for making sure the DNA is ready to be copied. The G2 checkpoint is in G2, and it's right before the cell divides. What happens in the G2 checkpoint, it's checking to make sure the cell is really ready to divide, that the DNA has replicated properly, and that the cells are big enough to make two new cells. And then the M checkpoint is called the spindle assembly checkpoint. Spindles are something that's important in mitosis, and we will learn about them a little bit more in the next few weeks. But once again, this makes sense right here, because before you finish mitosis, you want to make sure you actually have all the things you need in order to do mitosis. And so that's what happens at that spindle assembly checkpoint. So the idea with these checkpoints, once again, is just making sure the cell is ready to progress to the next phase of the cell cycle. There's another protein that's really important for regulation of the cell cycle and deciding if a cell can go to the next phase or not. This protein is called P53. It's really important for checkpoints. You can almost think it's like the master regulator of the cell cycle. So P53 can do a lot of things, but one of the things it does is it is really important in that G1 checkpoint. So if DNA is damaged, that damage in the DNA will actually be detected by P53. And then what P53 will do is it'll actually inhibit this cyclin CDK complex, and that'll prevent the cell from going to the next phase of the cell cycle. So you can think that P53 is almost like brakes on the cell cycle. It's all the time sensing if a cell is damaged or has mutations, and if it does, it's like, whoa, whoa, whoa. Let's slow down. Let's try to repair this damage, and if you can't, well then the cell is going to undergo apoptosis. And because of that, P53 is actually one of the most commonly mutated genes in cancer. So cancer is essentially uncontrolled cell growth. So the cells are just like speeding their way through the cell cycle super fast. So it kind of makes sense that in a cell that has lost control of its cell cycle, it's probably lost its brakes. P53 is like the brakes in a cell. So the way that you get cancer is just by unluckily getting a bunch of mutations in one cell, and the sum of those mutations is going through that cell cycle way too fast, which leads to uncontrolled cell growth. And so that is why the cell cycle is regulated with all these different proteins, and it's really complex because we have evolved to have this really careful checks and balances system to try to avoid our cells becoming cancerous. And that's some more details on how the cell cycle is regulated.